So the next slide speaks to another key pillar of our technology, which we call noise canceling. So we've described how the technology works in terms of detecting the presence of a target molecule by PCR. But there's also the challenge that Jacques had alluded to earlier, that in multiplex immunoassays, as the number of antibody pairs in a reaction or the number of plex or targets increase, the opportunity for nonspecific interactions between different antibody pairs also increases. In order to mitigate that, Codetta has developed the noise canceling assay approach, where the assay beads are programmed with both a capture antibody for a specific target of interest, as well as a target-specific forward primer. So a short single-stranded DNA PCR primer is also attached to the beads. So when the immunocomplex is formed and the detection antibody is bound to the bead through the target molecule, there's a DNA barcode tag that's unique and specific to each target of interest. And that unique DNA barcode tag contains a universal reverse primer, but a target-specific forward primer. And that target-specific forward primer sequence is complementary to the forward primers that are attached to the assay beads. So PCR amplification occurs only when there's the correct pairing of capture bead and detection antibody. And the next slide illustrates this how this noise canceling approach enables reduction of nonspecific signal. So in this case, with 3 target beads, target A has an immunocomplex formed with the correct pairing of capture and detection antibody. And again, because the forward primer on the bead amplifies the unique DNA barcode tag, that creates conditions that are conducive to PCR, and the resulting PCR amplicons are detected through the fluorescent intercalating dye, so a true positive reaction. But with multiplex assays, especially as you increase the number of plexes or number of targets per sample, there's opportunity for nonspecific interaction between antibody pairs. And that's illustrated with the red and green beads here, where if there's some low level of nonspecific interaction between a detection antibody for a different target, while that still may occur, there's no opportunity for PCR amplification because you don't have the correct pairing of forward primer to DNA tag present in the nanowells or microwells of the microfluidic chip array. Again, because there's only a single bead per well, there's only a single plex PCR reaction that can occur in each well. So any type of nonspecific interaction that occurs does not result in any PCR signal. And we show that experimentally in the next slide, where we have a 6-plex assay for 6 human cytokines. And the graph on the right shows a case where the noise canceling is not used. So in this case, just standard signal amplification was used to measure the binding of the target molecules on the beads. In this case, we just had a single target added to the sample. So 1 picomolar solution of IL-1 beta was added to a mixture of 6 beads, washed, and then 6 different detection antibodies were added and then amplified. And you see that we get a nice signal for where we expect to from IL-1 beta, but we also see, in this case, a fair amount of nonspecific binding from other targets that are not present in that sample. So this illustrates signal that occurs due to nonspecific interaction between antibody pairs, which obviously creates challenges when developing multiplex assays to control for specificity while still maintaining sensitivity. In the graphs on the right, we show the same reaction conditions, but in this case, the noise canceling approach was used to amplify the PCR signal. So in the case of the same experiment with a single target IL-1 beta added to the 6-plex assay, again, we see a nice strong signal where we expect to from IL-1 beta, but all of the off-target signal has been eliminated or suppressed, which we can then show holds true for the other 5 targets when they're added one target at a time. So by leveraging the noise canceling approach, we can develop multiplex assays that leverage the exquisite sensitivity of PCR, but maintain very high specificity through the use of noise canceling. This will allow researchers to leverage these unique capabilities to measure key biomarkers in a multiplex format with high confidence that the results are specific and that and while maintaining sensitivity to measure key protein biomarkers in serum and plasma samples.